In this lecture, I will introduce you to advanced instrumental analysis and data handling. Chances are, for most of you who have done Chem 211, most of the content, there will be a little bit of overlap, but nonetheless, it's important for me to start at this point. I think we all know analytical methods, can be classified into two groups. There's the classical methods of analysis, or the so-called wet chemistry. And actually that is the basis, of most of the Chem 211, where you do things like titrations, you do things like gravimetric analysis, where you use the weighing balances and so on. Now, classical methods of analysis preceded instrumental methods of analysis. So sort of this is the traditional way, of doing quantitative chemical analysis, where you can do things, like crystallization, precipitation, titration, gravimetric analysis, and so forth. Now, they're still largely used today for sample preparation. So even though we don't use them as often, for quantitative analysis, we still use them a lot, to prepare the sample for instrumental analysis. So classical methods of analysis still remain very relevant, today, but primarily, to get the sample ready for analysis, by the instruments. Okay. Now, in some cases, you're gonna see in today's way of doing chemical analysis, people have hyphenated or automated, the classical methods of analysis with instrumental methods of analysis. So sort of there's an interlink, where you can do things like extraction, becomes fairly automated, and that is connected, to instrumental analysis, such that things happen, in a pipeline. You can automate classical methods of analysis, and somewhat, they become instrumental methods of analysis, therefore. But our focus in this course is going to be different types of instruments, that are used for analytical chemistry. Now, instrumental methods of analysis, are things like, electrochemistry, chromatography, which, as I said, initially they used to be classical, but now with automation, they become instrumental methods of analysis. The wide range, of different types of instrumental analysis that we're gonna come across. Now, instrumental methods of analysis, mainly were adopted, as computations or electronics, developed. So they went line or shoulder by shoulder, as computers were being developed, so was the automation of classical methods of analysis for them to become instrumental methods of analysis. Okay. Now, the reason for that, is because instrumental methods of analysis largely rely on this paradigm. And I think those of you who did Chem 211, I explained that before. All instrumental methods of analysis, you've got a probe, and the probe is a source of the energy. And so the energy, can be photons, that's the most common, but they can also be electrons, they can be atoms, they can be ions, they can be even energy, all right, or rather heat. And so you can take, those forms of energy, and it heats the sample, and the chemicals in the sample undergo characteristic effect that is dependent, on the energy of impingement. And, the electrons, in the chemicals, undergo certain types of, interactions. and you can measure, the interaction, that results using a detector. So remember, these characteristic effects, can be very, very specific, for the molecule or the atom, at the investigation. And as such, you might be able actually to determine specifically what type of an analyte it is. And that's what you call qualitative analysis, where you're trying to find out what is present in the sample. Now, but also the response, can be proportionate, to the amount of chemicals that are present. And that is what you refer to as quantitative chemical analysis, where you're trying to determine, the amount of the chemical species that is present in the sample. Now remember, in most cases, this energy that is impinging, on the chemical, what you refer to as the analyte. The analyte, is a chemical species of interest that is present in the sample, whatever you are interested in. And so that analyte, of course, we all know a molecule or an atom, has got, electrons, around the nuclei. And so most of these energy actually goes into perturbating, those electrons. And those electrons, for example, they can be completely, dislodged, from the chemical, and that is called ionization. Or even, they can undergo, things like vibrations and so forth that you can measure. And all that, gives rise to different types of instrumental methods. So I hope that's clear. You can use different types of energies, and all of them, lead to different, types of instrumental methods. As I've said, most of the instrumentation, started to be developed, from the 1960s, which, is in line, to when computers, started to be developed. For example, in 1950, that's when, the very first, automatic computer, which was very, very big, you can see the size of a room, with only a RAM, of 1024 words or 36 bits. And, you can see that, over the last, 50 or so years, we've moved, from this kind of a gigantic, computer, to, laptops and even, iPhones, or smartphones in the last 20 years. And that has really influenced equally, the development of instrumental methods, which are completely, connected, to, electronic devices, and of course, computers as well. Remember, we're going to talk about this in the next slide. All these platforms that you're talking about, instrumental methods, really, it's an electronic device. And I'm going to explain, how you convert all these, physical domain into an electrical quantity, which you can connect with a computer, to do the measurements. Now, it's important, for us to remember, and I think I've mentioned that, the electrons, on the chemical undergo certain types of transitions, and those transitions, are the ones that, correspond to different types of instrumental methods. I'll give you an example. For example, in the case of using electromagnetic radiation as your probe, as your source of energy, which is the basis of spectroscopy. For example, if you use, say, ultraviolet radiation, we know it's fairly energetic and it causes electronic transitions, of the bonding electrons. And so for that reason, it can be used, for molecular spectroscopy, because of course bonding electrons are only present, in molecules, because the outermost electrons are involved in bonding. So you can use it, for molecular, spectroscopy, for example. On the other hand, if you look at something like an X-ray, you can see, that it causes, excitation of the inner electrons. So not the bonding electrons, but the inner electrons. And as such, you cannot really use it, for molecular spectroscopy, because it's penetrating, the bonding electrons and getting to the inner electrons. As such, you're gonna find in most cases, anytime you're using X-ray, as a source of the energy of the probe, then you can only use it, for atomic, spectroscopy. As such, you can see that you can use all these types of instrumentation either to probe atoms or to probe, molecules, depending, on how energetic, they really are. For example, if you use microwave, on the other hand, it's not energetic, enough to cause some excitation, of the bonding electrons, but instead, it causes, rotation of the bonding electrons. It causes rotation, of the bonding electrons, and so on. So I'd like you to take note of this. What happens, to the electrons, of the analyte, when they are impeached, by different energies, of the electromagnetic radiation. So it's important for you, to take note of that slide. But as I've said, not just the photons, which are the basis for spectroscopy, but the probe can also be electrons. It can also be atoms. For example, if you're using electrons, that's the basis often, for electron impact, the EI mass spectrometer. If you're using atoms, remember in our acronyms that we defined previously, fast atom bombardment, they can hit, a chemical species and cause, the perturbation of electrons, of that chemical species, that you can measure. And fast atom bombardment, is a good example of using that type of an approach to analyze proteins, for example. And so in this slide, I'd like you to take note, of the characteristic sample response, vis-a-vis, the analyte instrumentation. So as I said, if you take, the energy from the probe and heat a sample, a response, takes place. So the sample can absorb the radiation, for example, and that's the basis of spectrophotometry. The sample can, absorb the radiation, but also, excite the electrons, and then you get relaxation of those electrons, resulting into an emission of radiation. And that is the basis of emission spectroscopy. And there are many different types of emission spectroscopies that we're going to talk about. But also, the sample, can scatter, some of these radiation. And then, you can measure probably the transmitted radiation or even the scattered radiation, if you angle the detector, appropriately. And so if at all scattering happens, that type of, analytical instrumentation is often called Raman spectroscopy, which we're going to talk about in this course. But also other types, spectroscopies where you are measuring scattered radiation includes turbidimetry, which is very, very common, for, water analysis to detect dissolved solids and so forth. And also things like nephelometry. And the only really difference between turbidimetry and nephelometry really is the angle, at which, you position the detector. Now, other kinds of, characteristics that can ensue refraction, the light can be bent, and that is the nature of instrumental method. Also, things like diffraction can happen, where light is split, into its component wavelengths, and, the technique or the instrumental method that you get, can be something like X-ray diffraction, which is used to analyze, the crystal lattice structures, of, of geological materials and so on. I think one of the students was interested in geology and X-ray crystallography. It's a very, very common, technique, for analysis of geological materials. But you also know, the light, can be rotated, if at all, especially you're using, microwave type of radiation. And in that case, you can have a technique, such as polarimetry, which, or even something like circular dichroism, which is very, very important actually technique in organic chemistry, where you're trying to measure the so-called chiral molecules, or enantiomers, because I think the R and the L type of enantiomer rotate polarized light, in a different, directions. And so you can differentiate, between two types of enantiomers. Now remember, it's not just, spectroscopy. You can also have, something like electron analytical methods, where the probe in these cases is electricity. So you take electricity, and you push it, on the sample, and lots of things can happen: oxidation, reduction, and so on. That's the basis, really, of what you call redox electrochemistry, or redox chemistry, which is the basis of electroanalytical techniques. And you're going to cover a lot of these different types of electroanalytical techniques. As you can see, again, the energy in this case is going to be, electricity. Now, but also you can use thermal energy also. And the thermal energy is just something like, heat. You can have techniques, such as thermogravimetry as well, thermogravimetry, and this is where you use heat, or simply an oven, and you use that type of energy to induce on the chemical species. And thermogravimetry is a very important technique, especially for characterization of polymers, so that you can understand, their degradability or their thermostability. So very, very important, technique. People call it the TGA, thermogravimetric analysis. Very important, for polymer analysis. Now, so what's a general analytical problem, that we are trying to address, using all these different types of instrumental methods? Or maybe the question is, why do we have to innovate, so many different types of instrumental methods only for the purpose, of chemical analysis? And the problem lies in what you call the general analytical problem. Now, when you're analyzing a sample, often you're looking, for the chemical species of interest. And again, that is what you referred to as the analyte. Other than the analyte, the sample contains other types of materials that you're not really interested in, and that is what you refer to as the matrices. Essentially, they get in the way of detection, of your analyte of interest. And as such, because they are getting into the way, sometimes we refer to them as interfering species. They are making it difficult, for you to analyze your analyte of interest. Give you an example. If you have blood and you're trying to analyze the glucose in blood, think about all the other junk, that is present, in blood that you're not interested in other than glucose. There are proteins, there are red blood cells, there are salts, there are electrolytes, and so forth. And so all those other species, can be referred to as matrices, and they are actually the problem, in analytical chemistry. So that's often what you're trying to solve in analytical chemistry. How do I make an instrument, that mainly responds to my analyte, and the interfering species do not get in the way, of detecting the chemical species of interest? So what is the solution then, to interfering species or to matrices? And that's what analytical chemists really solve, in different types, of industries. But while you're doing food analysis, you can imagine, there's a lot of different types of chemical constituents, that are present, probably, in milk, for example. And maybe you're just interested in analyzing calcium in milk. And so all the other species are going to come in the way. And so you as an analytical chemist, you have to think, what is the best analytical method, that's going to help me, to analyze calcium specifically so that, all the other junk doesn't get into the way. So that's what analytical chemists do. So how do we address, this general analytical problem of matrix interferences, in sample analysis or in chemical analysis? So what's the solution to interfering species? Now, there are a couple of different things that you can do. Now, you can select or even develop, of course, in the research projects, we'll be getting, just to use, analytical method that's already developed, but you'll be developing, an analytical method yourself. So you can develop an analytical method, that is selective to the analyte of interest, meaning it's completely blind, to the matrices. And that's what often people refer to as sensors. And of course, there's so many different types of sensors, where the sensor is very specific to the analyte of interest, and it's totally blind, to the other types of interfering species, that are present. So often you have to look, for a molecular receptor, that is very, very specific, to the analyte of interest. And in most cases, maybe, the go-to, would be biological materials, such as enzymes. We know that they work, fairly well in terms of their selectivity. They are very, very selective, for certain types of analytes. Say something like glucose. If you take, glucose oxidase, glucose oxidase, only oxidizes, glucose and doesn't oxidize anything else. And hopefully, from that type of chemistry, you can find a way, to quantify, the amount of glucose, that is present. We're going to talk about that, later on. But the molecular receptors, for sensors are things like, enzymes. You can also use cells. Cells are very specific to certain types of analytes. You can use DNA. You can even use RNA. For example, the way they are trying to detect COVID-19, typically is, they get an RNA, that is very, very specific, to the COVID-19. And based on that, you can capture, you can capture the virus. You can capture the virus and then find a way, and we're going to talk about that later on in the instrumental methods of finding a way to quantify the chemical that is captured and hopefully, get an electrical signal, out of it, okay? And so if at all you use these types of sensors, people call them the biosensors. The biosensors meaning the molecular receptor is a biological material. DNA is a biological material. Enzymes are biological materials, and that's why they call them biosensors. But of course, we all know that there are so many analytes out there that it might be difficult, to find an appropriate biological molecular receptor. And in that case, chemists, sometimes, go for what they call biomimetic molecular receptors. And this is where chemists actually synthetically, manufacture, receptors that are very specific to the analyte of interest. In my research group, that's what we mainly do. We make smart polymers that are very, very specific, to capture certain types of analytes, and then we can do detection using different types of instrumental methods. And if at all you use anything other than a biological molecular receptor, people call that chemosensors. So one solution is using sensors, using an instrumental method that is selective. But of course, that's a problem, because that means, you have to generate or make an instrument for every instrumental method, and that's a problem too. So sometimes you want, methods, that can analyze multiple types, of analytes. So the other solution, is to separate, the matrix from the analyte. And that is the basis largely of classical methods of analysis. As I've said before, those classical methods of analysis, have been automated, now to become instrumental methods of analysis. So you can do separation using, and you guys have done this, I'm sure, in different courses, especially organic chemistry. You can do column chromatography, a painful process, very painful, trying to elute, analyte, from columns. You can do liquid chromatography or extraction. You can do solid phase extraction is another example of separating, the analyte using its ability, to get trapped, into a solid platform. Now, you can also do, recently, people have developed or miniaturized SPE into solid phase micro extraction, and we do that a lot in our research group as well. Now, you can also do things like crystallization, very common. You can also do things like precipitation, based on the solubility product equilibria that you guys talked about in Chem 211 and so on. Now, the problem of doing all this, is the fact that, these are very time consuming sometimes methods, and sometimes they're not very green, meaning you use a lot of organic solvents as well. However, you're going to see most of the chemistry out there, if I can say probably more than, 60%, of the chemistry out there, especially analytical chemistry, is based on doing this. So if you're hired in an industry, chances are, this is what you're going to be doing, in terms of analytical chemistry. You'll be trying to do separation and so forth. But as I've said, those techniques often have been automated, and there's a whole, progression towards that direction. And they've been automated, into things like gas chromatography, using different types of detectors. In this case, I've just included mass spectrometer. But you can also do, liquid chromatography, an automated form of column chromatography. You can also use capillary electrophoresis, which is an automated form of gel electrophoresis. So again, you can use that, to address, this analytical problem of matrices. In the last 20 years, or maybe 20 years now, people have tried to miniaturize, these techniques now into what they are referring to as microfluidic chips or devices. And sort of, this is a trend now, so that you can have very, very small devices that you can take to the field, for analysis. And sort of, my group, does a bit of microfluidic devices development as well. So, maybe just recap again, the generic component of all analytical instrumentation. So if I ask you, to design, or rather, to capture, the generic basis, for all analytical methods out there, they look like that. They've got a probe, energy stimulus, you've got, the sample that is being studied, and then you've got the response, or the detector, in this case, or transducer, in this case, okay? And I'm going to talk a lot about transducer. Now, you're going to see, these arrows are referring to, a very important component, that is missing, in this generic, sort of design. And the information that is missing, or the component, that is missing in here and in here, is what you refer to as, information sorter. And it's the goal of the information sorter, is to address, the bottleneck in chemical analysis, which is matrix effects, And you're going to find, this information sorter, and we're going to give it a name in the next slide. This information sorter often, are the components, that increase the cost, and they increase the bulkiness, of analytical methods. And so as an analytical chemist, as we are trying, to push towards miniaturized devices, these information sorters, are actually the problem, that we need to address. So for different type of instrumental methods, what are the information sorters? For example, assume you've got a spectrophotometer, the information sorter in that case, would be a filter. And of course, the filter in this case, is the idea is only allowing, selected wavelengths, to impinge on the sample and not the entire white light, to impinge on the sample. And later on, we're going to discuss why that's a big deal. Now, if at all you're doing AES, atomic emission spectrometer, the information sorter, again, is a monochromator. And the monochromator really is just an advanced form of a filter. And its goal, again, is to make sure that the analyte is being impinged by a monochromatic radiation. And the monochromatic radiation means it's got a single beam of wavelength. If at all you're dealing with, say, a coulometer or even a coulometer, the information sorter in that case, is a cell potential. If you're dealing with a pH meter, the information, remember this one is a sensor actually. Actually, it's a chemo sensor because you're not really using a biological specimen. So the information sorter in the case of a pH meter is actually a glass electrode. The glass electrode, is what is capturing, the hydronium ions of interest. They're going to show you in a couple of slides how that is translated, into an electrical quantity. If at all the instrument in question is a mass spectrometer, then the information sorter in that case is a mass analyzer. Remember us talking about, QQQ, the triple quad? Remember us talking about the time of flight, the TOF, and I was defining the acronyms. You heard me talk about the QTOF, quadruple time of flight, and so forth. All those, are examples of mass analyzers. And the mass analyzer, what it does, is to separate, the mass to charge ions or the masses of the analyte, based on their sizes, so it's sort of sifting, to separate, the masses based on their sizes. Again, you can see how it's addressing, the interfering effect. Now, gas chromatography, on the other hand, you can see the information sorter, is a column, is a chromatographic column and so forth. And as I've said, the information sorter, is often the major bottleneck in analytical analysis, as far as, making the devices, cheaper, selective, and so it impacts, a lot of the figures of merit or the performance, figures of merit, or the performance, of the instrument. And of course, later on, I'm going to talk about the figures of merit. But those of you from Chem 211, you understand what I mean by the figures of merit. Now, when you're talking about instrumental methods, you may find lots of people thinking of it, as a black box. Because think about it. Now, in most cases, we are interested, with a physical quantity or a chemical quantity. That's what I'm interested in. If I'm trying to analyze, say, blood, rather glucose in blood, what I'm interested, is glucose, which is a chemical quantity. It's a chemical quantity. Now, if at all I'm interested, say, in measuring, the heart rate, again, that is a physical quantity, the pressure, that's a physical quantity. And then you've got, these instruments, which converts for me, the physical or the chemical quantity, and it's converting it, into a language, that it can understand. And as we've said before, most instruments, nearly all instruments anyway, are electronic in nature. So how do you convert, or how does the instrument, convert, the physical quantity, maybe heart rate, or a chemical quantity, maybe, the glucose in this case, how does it convert it into an electrical signal, that it can handle, and then later on spit out at me the physical, the chemical quantity that I'm interested in. I do not care about the electronics, and really what I'm caring about is the amount of glucose that is present in blood. So you can see, there are multiple domains at play, when you're doing instrumental methods. I'm interested in a physical quantity. The instrument can only handle electronic language, but in the end, what I'm really interested in is a chemical or a physical quantity again. So what the instrument does, is, or the components of the instrument, what they do is to convert, these information, using the so-called data domains, is to encode the information and convert it into different data domains. So the physical quantity, can be converted to an electrical quantity, that the instrument, can handle. And then the instrument later on converts for me, the electronic quantity again into a physical quantity, which is what you're referring to as the analyst's domain here. So the instrument, in summary, has got these components, whose purpose is to convert, information, from one domain or data domain to another. So there are two types of domains. There is a non-electrical domain, and there is the electrical domain. So the instrument, is converting, the information into all those types of domains, okay? Now, the data interdomain conversion needs to be, is actually done, by what you refer to as a transducer, okay? So the critical components, within instruments, is what you refer to as a transducer. And the transducer, for example, the so-called input transducer, which is what happens first, the non-electrical quantity or the physical quantity, all right, is converted to an electrical quantity, so that the instrument is happy. That's the language the instrument understands. And then finally, you've got the output transducer that converts the electrical quantity, to non-electrical domain, so that the analyst is happy, and that's what you call the analyst domain. So you can see the data interdomain conversion within this so-called black box. But you as an analytical chemist, you cannot think of it as a black box, because you'll be required, to know what's the purpose of each component and how does it affect, the performance of the instrument, okay? And so in the next chapter, or lecture, I'm gonna give you a couple of examples, of how the different instruments, convert, all these information, between non-electrical and electrical domain, within the instrument.